Welcome to Time to Talk Science and Medicine, a new podcast designed to highlight translational researchers from Queen's University, Kingston, Ontario. The main goal of these podcasts will be to focus on the researcher, find out where they've come from, what motivates them, and how their research will change the world. We also try to find out about the person behind the dangerous idea, which makes their research so world-class. My name's Dr. Charlie Highmarch, and I'm a genomics researcher and faculty member at Queen's University.
And my name is Dr. Stephen Archer, and I'm a cardiologist and researcher here at Queen's, and I'm also the scientific director of the Translational Institute of Medicine. Today, it's my pleasure to welcome my friend and colleague, Dr. Lynne Postovit, who is a cancer researcher and, most importantly, the head of the Department of Biomedical and Molecular Sciences at Queen's, which is a big job, I can tell you for sure. And Dr. Postovit has a very interesting research interest, not only in cancer, but specifically in the construct of the DNA and its surrounding proteins, which are called chromatin, that prevent the DNA from being damaged and determine what parts of our genetic code are transcribed at what points in our life. She's made a career out of understanding the apparently chaotic process that regulates chromatin folding and unfolding that regulates gene expression. So, Dr. Postovit, thank you for coming into our podcast today. Before we get too deep into your scientific interests, I'd like to find out what motivated you to become a PhD cancer researcher.
Thank you, Stephen and Charlie. It's great to be here. So my motivation at the beginning was probably pretty accidental. So I started here as an undergraduate student at Queen's a little earlier than normal. I got out of high school quickly to be able to go to university. And I was considering physics and poetry. So poetry, not so great. And so entered general science here and started in doing physics and then decided to try out biochemistry and life sciences. A lot of my friends were doing that. And at the time, there was a show with George Clooney that was really, really popular that talked about medicine. So I thought that might be a great thing to do. And here at Queen's, we have an outstanding opportunity to have our students enter laboratories in their fourth year for a project, an honors project. And that was really my first exposure to research really at all. And it was during this time that I really fell in love with science. And it happened to be a laboratory that studied both early pregnancy as well as cancer. And so with that and sort of working through it, taking all the knowledge that I had, moving to the edge of that and seeing something new for the first time, I was completely hooked and decided to move away from George Clooney and medicine and to embark on a career in cancer research.
Well, that was our loss in medicine, but a gain for science. And it's sort of interesting. Most parents worry when their kid tells them that everyone else, all their friends are doing it, so they want to do it too, but they're usually not talking about physics and poetry and biochemistry. So how young were you when you escaped high school for university?
I was 17 when I landed in Kingston, so pretty young. At the time, most students were coming in at 19, so I was, I guess younger than most, but fully prepared. Fully
prepared, as are we all when we hit Queen's.
And I think it's interesting. You're back at Queen's now in the capacity of head of department, but you left Queen's at some point and did other things. How did that look?
Oh, so it's I guess an exciting time. So after Queen's, I stayed and did my undergraduate and then my PhD, and then moved to Chicago to Northwestern University with my mentor there, Mary Hendricks. And that's where I really got into my current obsession, which is cellular plasticity and this chromatin and epigenetics, and did a postdoc there. And I was there for only around 3.5 years or so. Exciting times, worked really hard and made some great discoveries. And I came back to Canada at Western first. So I was a professor there for 5 or 6 years where we continued to study cellular plasticity, epigenetics, so this idea of the chromatin, and understanding how cancers become more and more aggressive. And at that point, I was recruited out to the University of Alberta. And that was interesting because they recruited me as an ovarian cancer research chair. And I was never doing ovarian cancer research prior to that, but it's a disease that's really terrible. Over 50% of patients don't survive, or at least at that time weren't surviving past 5 years. And it's characterized by resistance and a lot of this plasticity. And so they brought me in, they said, no, this is great because we're going to get a scientist who studies other things to actually focus on ovarian cancer. And so we worked on that, continuing to look at this, and technologies got better and better. So more at the chromatin. So as we mentioned, the DNA and how it's put together and how it's responding to the environment. And then eventually I was called by a friend here, and they said, there's a job at Queen's as head of department. We think you should apply. And the rest is history. So I showed up around 5 years ago.
I remember that day distinctly. We were all very happy. I think I might have even been on the search committee, but I can't attest to that.
You
in
were. You
I was.
were.
Okay,
Yeah.
Well, then I'll take credit for you being here.
That's quite a homecoming, though.
Yes, yeah.
One thing that in preparation for this, I was reading and talking to you, reading what you researched and talking to you. And one of the things you mentioned was epigenetics and chromatin or chromatin, whichever pronunciation we're going to stick with. But one of the things that's interesting that I learned in my reading was that we have 2 meters of DNA in our cells. So in one cell, 2 meters long of this DNA, but it's all wrapped up in a nucleus, which is microns in size, 1/1,000,000th the size. And is that what chromatin is doing, is winding up that DNA?
Yeah. So the chromatin is the DNA and the proteins that are basically like little beads that the DNA is wound around. And how it works is that there are modifications. So it's almost like putting little spikes on the beads or molecules of some sort that allow them to move farther apart or closer together. So as they move apart, it opens up, and then the DNA becomes available to be made into a message that can eventually make a protein, and otherwise it can close really, really tightly together so that those messages can't be made.
So the doors are opening and doors are shutting all the time as part of a dynamic.
Precisely. So if you think about it as doors and in a controlled situation, so a normal cell, exactly the right doors and windows are open. It's all under control. The breeze is coming in. Everything is great. And then we're thinking of cancer where those are moving, opening and closing really quickly. You never know what's going to be open at one time or closed at another. And it's fairly unpredictable.
It is interesting that the other way to look at it, I guess, is now that you mentioned this analogy to winding around chromatin, is that people that are tightly wound, none of their DNA could be transcribed. It would all be locked down by their chromatin, not accessible to transcription factors. So next time we use the phrase tightly wound, I will think of someone who's completely transcriptionally repressed.
Yes, that's right.
We'll move on from that. So on this show, we like to ask researchers about their dangerous idea. And by dangerous idea, we're really talking about the concept where, if true, it would change the landscape of the field and it would change the direction that people think about the field. Can you tell me about your dangerous idea, please? So
I guess our dangerous idea, because we're working on this with people across the globe, as most of these, is that very advanced cancers, so the ones that resist therapy and spread and that patients ultimately die from, are due to completely discoordinated epigenetic regulations, so absolute chaos. And in cancer, we think a lot of targeted therapies, and there's been great examples of ones that work really well. So to BCR-ABL as an example. So this is a mutation that occurs in some blood malignancies that you can come in and target and really take away the cancer. We're basically saying there isn't one target or one node, and it's really this ability to move and to change, to have the doors and the windows opening randomly and unpredictably that's driving the cancer. And so we think that if we can figure out how that happens, what's allowing it to happen, or perhaps how to just slow it down a little bit, we may find new therapies for these cancers, which we otherwise can't do much about.
I mean, it's a really complex orchestra of events that are happening in every single cell, normal or otherwise, that's happening simultaneously. And these cells are all talking to each other. So what kind of strategies do you use to kind of realize your dangerous idea?
So this is where it's been really exciting in the last several years. So we use a lot of techniques that involve stressing cells. So we look at hypoxia to try to mimic that and push things in one direction. But importantly,
hypoxia means changing
low oxygen levels, which are really, really common in almost all solid tumors because they grow so fast the blood vessels can't keep up. And so we've also been using tools that allow us to see cells one at a time. We use genomics, so you're a geneticist and do genomics as well. And this is basically being able to look at every transcript, so every one of those codes that's going to make a protein in a cell, all of the chromatin and all of those marks that are allowing things to open and close, the actual DNA itself to see which windows are open and which windows are closed, as well as all the proteome. And we can look at this at the same time now at a resolution that's unprecedented. So we can see in snapshots over time how the chaos is forming and where it's forming, so that we can get an idea of which features are most important.
So I get it from this that there's not one thing wrong in cancer, which is why cancer is so successful at causing harm to human beings. There's many things wrong. And you mentioned several times about chromatin and how that's important in this. And my understanding of chromatin is that one of the ways its opening and closing is regulated is through the process of methylation, where chemical groups are applied to the chromatin that either make it wound tight or wound loosely around DNA. And when you say many things are going wrong, are those many things related to methylation of chromatin, or is that something else?
So one of the things is actually methylation to chromatin. And so these are the little beads themselves that can be methylated or demethylated, as well as even the code, so the DNA in between. And that can either activate a gene or inhibit it, depending on where those methylations are actually occurring. And what's interesting, and why we were really interested in also looking at low oxygen, is it changes the whole state at which these the enzymes, so the proteins that can put down these marks, work. And so they need oxygen in order to take the marks away. So what you end up having in something where there's low oxygen is an accumulation of these marks everywhere. And so you're allowing a sort of dysregulation to occur.
It's kind of like the chromosomes are like electrical wire, and this is like the coding on the electrical wire. And can you actually see that with a microscope?
Actually, you can now. So there are microscopy techniques that are extremely high resolution. So we can see down to a molecule. We can then label protein as well as DNA and be able to see where they are. In that, we can come in with antibodies and see where those are as well. And then with our sequencing technologies, we may not be able to see it exactly, but we can map out the entire genome as well as all those little beads and precisely tell you where they are and which beads have a modification and which don't.
So there's this complex orchestra of events that is happening. Is there any kind of grand master plan that's running of this? Because you mentioned the word chaos, and that's the opposite of a grand master plan. Is that what's happening in these cancer cells?
Yeah. So we're really thinking that in advanced cancers, they've gained the ability to be chaotic. So a normal cell will die. We call it apoptosis, or maybe another kind of cellular death, if chaos occurs, because it's just not supposed to. It's a defense mechanism.
The cell recognizes it and shuts it down.
Absolutely. And in cancer, it somehow has gained the ability to overcome that, so can sustain this chaos. And so that, instead of the cells working together to have a function, a collective function, one cell is doing one thing very selfishly and the one next to it could be doing something completely differently. Doesn't happen in normal multicellular systems. And so cancer is unique this way. So it has to have something that allows it to survive that.
So this is probably overly simplistic, but on the theme of the dangerous idea, would the dangerous idea practically translate into something that regulates methylation of chromosome, which deals with the chaos? And if so, how does it know whether to close the door or open a door? Because both are happening at the same time in a cancer cell.
So that's the $1 million question. And it would be probably, yeah, there you go. It's our next grant we should do. But I think it's about sort of bringing it all down to a level to create a vulnerability of sorts. So you could imagine, for example, if we have a lot of methylation accumulating, because those proteins that take the methylations away no longer work, we could actually have an inhibitor that prevents the methylations from being laid down, bring it to a normal level, quiet it down, and come and hit the cells when they're down and a little less able to move in different directions.
And you were indicating in an earlier conversation that some of this is non-random, that these methylation things that might affect the condensation of chromatin might occur in one place and not another. So there may be ways of targeting the genome, at least in the future.
Yeah, for sure. So this is sort of an area of research that's relatively new and is increasing rapidly in our knowledge because of these types of technologies. And so we know just in these proteins that are going to lay down those marks onto the beads, that there's different types of them that go to different places. And so we're really asking the question now of how are they differentially regulated and which ones are the most important for allowing some of these changes to occur.
So I'm going to take you back to high school now. So remember your 17-year-old self. So I'm going to ask you to explain your current, somewhat more than 17-year-old present self. Tell your old self in high school terms, what you do and why this is important.
Okay, so this is a challenging one.
Self.
Yeah. Hello, self. So if you can envision cancer and advanced cancers as not just one disease or even 100 diseases, but 1,000 of them or 1,000,000 of them, with different cells moving in different directions all the time. So everything that they learn to be during development, so a skin cell learns to be a skin cell, and a neuron, a neuron, and a cardiomyocyte, or a heart cell, they've just forgotten that, and they can move between these particular fates. And that ability to move means that if we come in with a therapy, we may be able to take out 999 of 1,000 cells, but that one cell still remains, or maybe there's 10 cells, and they can move even more. Their whole thing is the ability to move around and to evade therapies, evade the immune system. And what we're really trying to do is understand, how is this even possible? So we have a system, a body, where everything is coordinated, and it has to be coordinated to work really, really well. Yet in this cancer, it's undone all of that. So what are the keys to that? How can we come in and prevent it from happening, or at least quiet it down for a bit so that we can come in and target it and take out all 1,000 cells?
So cancer cells are shapeshifters, and
that's brilliant. Yeah. Shapeshifters is a great way to think about it. Yeah.
So thanks very much for the kind of the detailed explanation, but also the kind of the high school education. You obviously have a broad portfolio of work, and I guess just to kind of pin down what your latest discovery or a discovery you're particularly proud of that you think has kind of traction in the community. Can you tell us about one?
So one discovery we're really excited about is getting to this notion that all of these systems in a cell are working together to enable chaos. And so in this particular discovery, we found that low oxygen, as I described, so hypoxia, can actually change where genes begin to start to make their code. And they do this by moving those beads around, that chromatin around, and changing that methylation mark, which is really interesting. What's even more interesting is that it works with how cells make a protein. And so to make a protein in a cell, it needs a lot of energy. So we call this ATP. And so when we have things like hypoxia, where we make less ATP or there's less nutrients, the cell reacts by downregulating or reducing all of the proteins that it can make, while at the same time changing that machinery that makes proteins to make those proteins that can allow the cell to survive. And so what we found is that these beads that change the code don't necessarily change the protein that's made, so that's going to end up helping the cell, but actually change where they start in that little bit at the front of them that tells them how they can fit onto the machines that make the protein. So really, we've discovered that the epigenome, so this, the DNA on a bunch of beads, is actually dictating the proteome, or what proteins get made under times of stress. And this is important because it's important for the adaptation that I described. So one of the things that seems to be happening because of this is a switch from using oxygen to make energy to sugar to make energy. And some of the proteins involved in that are actually targeted by this mechanism.
Yeah. I'm surprised that Stephen hasn't mentioned mitochondria yet, but
I'm just salivating. Yeah. Actually, I was going to go a slightly different direction, because you and I are co-PIs on a large Canada Foundation for Innovation grant called TIMECORE, the Translational Institute of Medicine TIMECORE. And this is going to give, and Charlie is an instrumental partner in that grant, which is about $15 million. It'll be coming to Queen's soon. So with all this cool work you're doing on the epigenome and chromatin and cancer biology, are you getting some new toys out of this deal that will allow you to take your research to the next level?
Absolutely. I think probably everyone doing fundamental biomedical research here will have great toys to answer this. Some of them, for example, allow us to see what the cells are making in a tissue, so where they are and exactly what they're making at any given time with single cell resolution. So it's the state of the art in that area. I hope to work with you to maybe start to see some of these things a little more with these fancy microscopes that allow us to see how structures are changing and where they are in organelles. So the bits and pieces of the cell that do all of the work. And we also have an amazing animal facility that will be built into this. And a lot of the work that we do, and I get very excited about this basic fundamental stuff, is geared towards finding new therapies for patients. And so we have models to be able to test new therapeutics, and to see whether they may be viable to move forward into the clinic.
It should be fun when we open up all those boxes and get our Christmas gifts.
Absolutely.
Yeah, absolutely. I think the answer also kind of underpins the importance of kind of investment into science in Canada and how important it is for you can have dangerous ideas, all day long, but unless you have the tools and the people to realize those ideas, they come to nothing.
Absolutely. And you really need an ecosystem and everything together. And we often focus at the endpoint, so the things we're about to bring to trial. And I've had the fortune of doing these crazy ideas or brave ideas, and then having those evolve into new therapeutics that we've tried out. And if you don't keep having them, you don't have the tools to keep pushing it and testing them, you can't have new things to bring forward. So it's special and important.
So becoming a department head is a huge success, and being the co-PI on TimeCore is a huge success. But I know that every scientist has bumps in the road, so things that don't quite go as planned that you have to persevere with. So what, Dr. Postovit, give me an example of one of your bumps in your journey.
I think one of the bumps was actually early on, and it's been sort of resolved over time. And that was that the idea of this plasticity in cancer, so that the chromatin is moving around and in different states, was sort of contentious when I entered doing this work in the early 2000s. And so the idea that a cell type could move from one fate to another, or even completely, what we say, de-differentiate, was difficult to see, difficult to prove. And so there was a lot of skepticism at the time. So we're writing papers about it and trying to push this through. And it was actually very difficult. And that has been overcome again with these technologies where we can see that this is happening. With single cell resolution, we know that we can tell a cell that looked like something one day looks like something completely different another day, and that they're actually moving back and forth. And so we were inferring this and testing it back then with skepticism. And now I think folks around the world are seeing that this is actually happening definitively.
So your problem, your bump in the road, was actually getting buy-in from the wider scientific community for the idea.
Yeah. It makes it a lot harder to, get the funding to do the work, et cetera, et cetera. So that was a bump, but I think I was pretty passionate about the concept and idea. And so you go up the hill and
do you think, just again, coming back to the funding issue, do you think that kind of enough resources are being put to high risk ideas?
I don't think there are enough resources being put into fundamental research, period. Whether it's because it's too abstract or not, timely right now in our current state. The more that we can have for these great ideas and exploratory concepts, the better.
I think it's true, sitting on a CIHR panel, and I know you do too, and CIHR, Canadian Institute of Health Research, is, of course, a major source of funding in Canada for researchers. But there's sort of two problems. On one hand, if it sounds like a crazy idea, like the plasticity of cells, they can shift their molecular structure and thus their pathogenicity. That may sound crazy for a while. It goes from can't be true to must be true to everyone knows this is mundane. And at all points in that cycle, it's difficult to get funding. And I think right now, I believe our funding rate is only 18%, so less than 1 chance in 5 of getting a submitted grant funded.
Mm-hmm. It's low. And I always point out that this isn't just anybody applying for grants. So these are people with established track records and strong programs and working hard at our university. So this isn't just anyone off the street with an 18% success rate. This is the very best of our researchers struggling with this sort of low yield endeavor. Yeah.
And it's also interesting because they're not only doing their research if they get the funding, but they're also teaching our undergrads and our graduate students and basically making universities work in addition to doing their research. So it's a...
Yeah, 100%. So it's generating the knowledge that we need to make the world better, but then also the people who will make the world a more amazing place and contribute to our economy and to discoveries and to caring for people. And so, most of our research grants actually go to supporting those trainees in the end, and amazing scientists. So it's difficult.
So within the context of your research and your field, what's next? So this is a crystal ball question, and the answer is what's next for the wider field but also for your own research.
I think there's two things sort of interrelated. One are the computational methods, and this could be AI, artificial intelligence, to start making sense of how different pathways and different systems in a cell are actually working together to ensure chaos or to enable chaos.
Because of course, in the last 20 years of what we call the post-genomic era, since the first draft of the human genome was produced, a huge amount of data, an exponentially growth of data has happened. So do you think AI is going to help with that?
Can I just interrupt and just say, have you guys not seen The Terminator? I mean, there is nothing good will come of that.
I watched Jurassic Park and that worked out well, right?
I think what the oh boy, what the AI will do that's not so scary is start to see patterns that are there that are much more difficult for us to see just because there's so much of it. And it's within those patterns, and I'm not talking about individual genes or individual proteins, but rather whole systems that are moving that'll build new hypotheses for us to test. And the trick, I think, is going from a scientific method and, a Sir Bacon-esque method where we, put everything aside and then really focus so that we can answer a yes-no hypothesis, taking that and then transforming it or adapting it to be able to consider multiple parameters. So we're really good at considering one or two. This is really about considering whole homeostatic shifts. And so the movement of numbers of parameters. So this is a problem of mathematics and statistics, probably physics, and going back there, maybe a little bit of poetry, but really a transformation of how we think and design experiments. So to take all that data and run really meaningful experiments that allow us to test that.
Well, while you're thinking of a word that rhymes with chromatin so you can resurrect your career as a poet, I want to thank you for taking the time to come in and speak with us on the podcast today and tell us about your fascinating research. And also, I know firsthand the impact you've had at Queen's as a department head, which is an often thankless job. So on behalf of your colleagues, thank you for what you do, Dr. Postovit. But now, of course, we'd like to demystify you and change you from a glorious researcher into someone who's just going to answer some random stupid questions that Charlie and I have made up. So I'm going to let Charlie have the first kick at the can.
I'm going off script for this one, and you mentioned poetry twice in this podcast already. So an opportunity for some original work, perhaps.
Right now, no, can't do it.
So do you still write poetry?
I do. When I have time. So I think you have to be in a state where you can almost see things. So just be able to visualize a scene or a time or a place, and it's a lot of deep thought. And so when I have some quiet time, I often do. So I'll take a book and sketch out and write out these types of things. So it's not a lot of time right now with everything going on, but certainly on vacations and times off. And sometimes I'm fortunate here in Kingston to live close enough that I can walk to work along the lake. And sometimes I get so inspired by the sunset or the way the water looks or just a sound or a memory, because I was also a young person here, that I have to stop and write something down.
So you get into the zone and then
you need to be in a zone. Yeah, you almost have to be... it's a bit of transcendence. So you're kind of there, but not there. You see something else when you're writing. So
essentially poetry with the sort of parsimony of words to express big ideas. It's almost the opposite of science where there's 1,000 words to express a simple idea. Poetry is a few words to express a very complex idea.
Absolutely. So, and I will admit there was a time in almost all of my papers where there's one line or one sentence that's more poetic than functional.
Okay, on to your next skill testing question. So superheroes. So what type of superhero would you be if you could pick from any of the existing superheroes that you're probably familiar with? Is there one that you think is you?
Oh my gosh, I really don't know. So I certainly not something like a Superman or Superwoman or anything like that. It would be more of a kind of in the background trying to make things better some way, but I can't tell you. I don't know.
Well, that's your homework. We'll let you.
I've got to think about it deeply. I don't know.
Typical of recordings, after you go away, you'll immediately say, I know who I am.
Yeah, who am I?
So I know that you enjoy cooking, so kind of it takes care of part of this. But so what is your favorite meal, and can you cook that favorite meal?
Oh gosh. I have a number of them. So probably my favorite meals are the ones that make the people that are most special to me really, really happy, because I love them, they love them, and there's just excitement. So right now it's probably a lobster bisque with some kind of a meat or something that goes along afterwards. But usually the lobster bisque is pretty filling. So I really love to make that, and it's my son's favorite thing. So yeah.
That sounds delicious.
It's actually pretty awesome.
I'd love to try it.
Yeah.
So you mentioned in some conversations we've had previously about mentors you've had. And so my last question for you is, who's your favorite active scientist that has positively influenced your career?
Holy geez.
I know that's tough. No, not that we don't want to make people second and third favorites, but just even if you think here locally at Queen's, like, is there someone here that influenced your career?
Yeah. Well, I mean, I did do my PhD here, so I always do a shout out to Charles Graham. So he was in anatomy at the time and in DBMS, so I'm now his boss, which is interesting. But it's really Charles who saw a young girl at the time who came in and was looking for a research spot. I had a hard time thinking linearly, so I think I probably said a lot of things that didn't make sense to anyone but me. But he saw something and took a chance and took me into the lab. And then he allowed me to really thrive as I am. So I am an independent thinker, not really great at being told what to do. And he allowed me to explore and to make those discoveries. And I have to say, there are those moments when you're in the lab and you look down and you see something for the first time on the planet, maybe, or you recognize it at least for the first time. It's exhilarating and I guess addictive for me. So he allowed me to do that, and probably is the reason why I went into science.
On behalf of Dr. Archer and myself, I'd like to thank you for taking time to get to know Dr. Postovit, one of Queen's University's translational researchers, and finding out how her research will positively change the face of cancer therapeutics. If you've enjoyed this episode and would like to know more about Queen's University translational medicine researchers or share this podcast, you can search for Time to Talk Science and Medicine wherever you normally look for podcasts. On behalf of Dr. Archer and myself, thank you very much, Dr. Postovit.
Thank you.